All right, good morning, afternoon, or evening to everyone joining us today. My name is Jessica Taylor, and I'm here at the Canadian Centre for Evidence-Based Conservation at Carleton University in Ottawa, Canada. So as you may know, we created this webinar series to replace the cancelled in-person Environmental Evidence Summit that was supposed to be here in Ottawa this past June. While we wish we could have all been together this spring, this series has allowed us to reach hundreds of more people than we had originally planned, and given us a way to connect with you all over the next year through monthly sessions. We've got an exciting session today, so we'll get started right now. On behalf of CE and our team at the Canadian CE Centre, welcome to our session on evidence synthesis technology, which will be chaired by Neil Hadaway.
Thanks very much, Jess. Sorry, I was struggling there a bit to turn off my microphone. Hopefully everyone can see my screen. So we've got a really exciting list of presentations today. We're really honoured to have a large number of people who are real experts in the field of evidence synthesis technology. If you have any questions as we go, each presenter will have about 10 minutes to present, followed by 2 minutes of questions, depending on the timing. And then we have a short 12 or 13-minute question and answer session at the end as well. Unfortunately, some of our presenters have to leave a little bit early, but any questions that come in the questions tab rather than the chat tab, please. We will either answer them live or we'll answer them as we go during the webinar, but we'll also export them and ensure that they're answered and put them online, along with the recording and the presentation files at the end. So here's our list of presenters today. We have a range of people from different disciplines, different backgrounds, and from all over the world. So we're incredibly thankful for that. We will put up these details if you have any questions for people specifically as well, if you want to contact them at the end. But we will start off shortly with my presentation. So I'll just transfer over. Hopefully you can all see that. So my name is Neil Hadaway. I am a senior research fellow at the Stockholm Environment Institute in Sweden and the Mercator Research Institute on Global Commons and Climate Change in Berlin. I have a background in conservation biology and ecology, but I've recently been doing systematic reviews across a number of fields, including international development, social welfare, and some in health, mostly in the field of environment and sustainability. The presentation that I'm going to give to you today is on behalf of my co-author, Martin Westgate, is introducing the evidence synthesis hackathon. And I'll just spend 10 minutes introducing the hackathon, telling you a bit about the background and what we do. I wanted to start off briefly by just giving some acknowledgements to our funders, our generous funders. Mistra EVM, the Australian National University, the Africa Centre for Evidence, the Alexander von Humboldt Foundation, the University of New South Wales and the R Consortium, all who've given generous funding towards the Evidence Synthesis Hackathon events. So before I get started in talking about the Evidence Synthesis Hackathon, I just wanted to spend some time thinking about what we mean by evidence synthesis technology. And broadly speaking, everybody knows really that technology is a collection of techniques, skills, methods, and processes. And that's probably a slightly more broad definition than you might have thought. It sort of includes more skills and methods and frameworks, as well as tools and software. But what you define technology as kind of depends on when you ask. So at one stage, systematic review was a novel technology. But in general, we're talking about tools to support evidence synthesis to overcome the challenges that we face when we conduct evidence syntheses. And they sit on a spectrum from computer assistance, which might be termed technology-assisted reviews, where technology is being used to support capacity development or training, to perhaps improve or facilitate record keeping in systematic reviews, increase the efficiency or transparency or rigor of reviews. And then on the other end of the spectrum, we have what might be termed technology-driven reviews, where certain processes are replaced by a computer. Can you all see my screen, by the way? I hope so. Just had a question from one presenter who couldn't. I will carry. Yes, I think most people can. Great, thank you. So why is evidence synthesis technology important? Well, we all know if we're working with systematic reviews or other projects that we're often dealing with a lot of data management in a review over a long period of time. We deal with big, complex and messy data. Often it's hierarchical or interrelated. There's often issues around version control and multiple file types, particularly in working with a team, and ensuring that we have backups and that access and archiving of files is secure and reliable. It starts to get quite complicated. Along with that, particularly this year, we're all very aware of the challenges of working remotely in a team. And in a systematic review or a systematic map, we're also trying very hard to be as transparent as possible and apply open science principles to evidence synthesis, what I call open synthesis. And then pragmatically, we're also dealing over recent years with an explosion of literature. So this figure shows recent environmental evidence, systematic reviews and systematic maps, and the search results from each review. Each line is a review, and it shows the number of search results per year. And in virtually all cases, there's almost an exponential increase in literature that we're having to deal with. So as time goes on, we're facing more and more reviews. And so technology is really important to help us deal with all of these challenges. There are a number of tools that are available to help us. I've just put a few on the screen that are cherry-picked from what I'm aware of, that help from everything from planning, managing the review in specific systematic review management tools, helping us to search, deduplicate, screen, extract data, and critically appraise our studies. Some of these are even pushing towards automatic extraction of data and automatic critical appraisal. And then tools that help us with synthesis and updating as well. So some of the problems that we face in evidence synthesis technology is that we are aware that there are many tools. There are an increasing number of tools available. If you're interested, you can check out the Systematic Review Toolbox to find out just some of those tools. But many of the tools are paywalled, so you have to subscribe or pay to access them. And often those that aren't subscription-based or paid are not supported in the long term. There's also a lot of redundancy with many tools that aim to do the same thing, and also very limited interoperability. So if you are using one tool, it's very difficult to move through the systematic review process from one tool to another. And another issue is that many of the tools are not validated, or they've been validated by the developers. So we're not entirely sure that they work consistently across reviews in different contexts. There's a group called the International Collaboration for the Automation of Systematic Reviews, and they set out the Vienna Principles, which aim to improve the way that technology is developed to support systematic reviews. They state that there should be the ability to work with different tools at different stages of a review, and that the tools should be interoperable, allowing people to move between them. They also state that there needs to be collaboration across the community of people who are developing tools, so that we can cover all stages of synthesis in all different contexts. They also call for the code for these tools to be open source to improve collaboration. And they also believe that there should be standardized validation of tools using standardized data sets to show that they work consistently. So Martin and I recognized this challenge with evidence synthesis tools, and we decided to develop the Evidence Synthesis Hackathon. And the hackathon aims to bring together an interested group of researchers, practitioners, and coders to discuss and develop new open source technologies for evidence synthesis applications. And our mission is to support development, testing, and promotion of new software tools and workflows, to build networks and build capacity amongst researchers, practitioners, and developers, and to advocate for open synthesis, that's open science principles applied in evidence synthesis. Our goal is also to create workflows that are as open as possible, that are reproducible, and that are based on the best available technology and methods, and that are also widely supported by the community. So our hackathons are, or have been to date, typically 3-day interactive workshops, which up until this year were in person. We've had at the most recent hackathon event a separate coding stream, which is focused on the production, development, and testing of software, and a discussion stream for evidence synthesis experts, in particular, to discuss new frameworks and ways of working and ways of thinking about evidence synthesis. We run these hackathons in a slightly different way to normal hackathons in that they are non-competitive. We encourage people to collaborate across different groups and to move between the groups or between streams so that they can use their skills in the most appropriate way. And we aim for a balance of backgrounds across synthesists, decision makers, and people working in business, across career stages, geographies, the global south or low and middle income countries, and diversities as well. And this is our most recent physical workshop, which was in April in 2019. And so far we've had 4 workshops, or 3 physical workshops, one in 2018, a follow-up workshop which focused on just one tool, and then our biggest event so far in Canberra with 46 participants in 2019. This year we have been running a slightly different process where we have long-term projects that are going on in the background throughout 2020. And we also aim to run a conference on evidence synthesis and meta-analysis in R in January 2021, so watch out for that. And in 2021, we're also hoping to have a physical workshop, if possible, sometime in the Netherlands. And so far, we've had over 100,000 euros pledged for just 2 workshops. In our discussion stream, we've had quite a lot of success in forging ahead with discussions and making those public. So we have a paper in Nature Ecology and Evolution thinking about a new ecosystem for evidence synthesis. And we have another set of papers and projects that are focusing on bibliographic database limitations, on trying to make primary research as synthesis ready as possible, and assessing evidence syntheses in the field of environment to see how open their data and code are. And then we also have in the coding stream a number of projects that I won't go into detail on because some of them are being discussed here, but you can find out more on the projects. And many of those are now fully functioning tools. And there are a number of ways to get involved in the hackathon. You can propose a coding or discussion project, and you can contact us with those suggestions or meet us on social media. You can use and test some of the tools and help provide feedback or point out bugs or how to improve them. You can apply to join a future hackathon or one of the ongoing projects. You can check what projects are going on at the moment on the ES Hackathon website. You can offer to co-host a hackathon, and you can help us to fund a physical event. We're particularly interested in supporting lower middle income countries. So that's it for now. Hopefully that gave you a bit of an introduction to the hackathon. You can find out more at eshackathon.org. Okay, so over to me to field questions. Does anybody have any questions? You can feel free to pop them in the chat. We've got a couple of minutes before Martin takes over. If not, feel free to post a question I can answer throughout the session. Otherwise, I can perhaps hand straight over to Martin. Jess, I don't know if you want to hand that over. Great. So as an introduction, Martin is a senior advisor with the CSIRO Atlas of Living Australia and co-founder and coordinator of the Evidence Synthesis Hackathon. So Martin, over to you.
Lovely, thanks, Neil. I'm just going to hope this works. Someone yell at me if you can't see my screen or hear me properly. Yeah, so thanks for that introduction. Neil's going to give a good overview there of the work that we've been doing over the last 3 years with the hackathon. And what I'm going to talk about is a specific project that's come out of that, which we're calling Metaverse. And it's basically an attempt to bring together a series of tools in the R ecosystem. So I'm going to presume that most people are aware that R is a statistical programming language. Has a lot of users these days. It's very widely used in academia, but also more broadly. And we're trying to have an impact in that space because of its wide usership, but also because it's open source and free and has some advantages therefore for users. So I'm going to go through what motivated us to build Metaverse, what it's used for, where the development of this is going, and some things we're not going to do, because I think that's important to limit ourselves a little bit as well, in that you could work on this stuff forever. And all the stuff I'm going to talk about are products from various evidence synthesis hackathons, including the main package Metaverse itself. So why do we think that yet another R package is needed? So for those that don't know, R is a programming language, and you can extend that language by building what's called a package, which is a set of tools that work together in a consistent way. And there's a central repository, an archive for those called CRAN. There are now over 12,000 packages on CRAN. So there's a lot of those have relevance to some part of the evidence synthesis process. What the problem is that when you have that many, it's hard to find them. So, and to clarify that a little bit, there are things like there's a subsection of the website that's called a task view that enables people who have an interest in meta-analysis to find in great detail specific tools. But more broadly, if you think about the process of doing a systematic review, there's a whole lot of stages to it that aren't meta-analysis. How best to find literature, how best to screen it, tools like getting text out of PDFs or transcribing results from diagrams in papers. These are all very different sets they require very different software to solve them. And so what we found when we ran our first hackathon in 2018 was that we tried to say, well, what are the software packages in R that you would use for a systematic review in general? And we couldn't narrow it down very easily. It's very difficult to define specifically what, not necessarily what the best ones were, but just what a cohesive set of packages looks like. There's some things that are very general packages that are useful, and should we include them, or things that are too specific, which includes some critical information. So, and critically, we tried some computational approaches to do this, mapping the R ecosystem, and that didn't work either because they, for various reasons. So what this meant was that the R package ecosystem was effectively a microcosm of all the problems that Neil was raising earlier on. People tended to develop their own packages independently of one another. It's limited integration between them. There's duplication, but there were also large gaps where there were things that we would think that R would be well suited to, and yet were not available in the R package ecosystem. And so we built this Metaverse project as an attempt to find and integrate R packages that could work well together. And so what it looks like right now is there are 8 packages. These are all the hex stickers or logos for them, with the exception of Metaphor, which is just a Metaphor doesn't have one just yet, but may do in future. So this is just a place filler. But basically, going from left to right, this is roughly the stages that we thought we could support well from a systematic review now. So the latest package we've developed is the first one here, called Synthesizer, which is for importing bibliographic data to R in a clean way. It's not the only package that does that, but it tries to be as versatile as possible and import data from a range of formats and locations. The next package, in terms of stages of the systematic review you might be interested in, is Lit Searcher, which Eliza may talk about later on as the lead developer on that project. Which is for search term optimization. My own contribution is RevTools, which is for article screening, but particularly for visualization of text data. There's a gap here that I haven't shown, which is about, once you've done your screening, how you extract data. We do have meta-digitize, which is for getting data out of plots, but it's not a general purpose piece of software for transcribing all the data you might need for a meta-analysis. So there is a conceptual gap there. Proceeding on from there, the metaphor package is part of Metaverse, which is, as many of you will know, a standard piece of software for meta-analysis in R. And then finally, there's the RobVis package, which is for visualizing risk of bias results. So between them, they cover quite a large proportion of the range of tasks we think you'd want to do during a meta-analysis and that R can currently support. So we're adding to this, and we're doing that building on software that came out of the Hackathon series. So the first of these is some work that Neil and I did a couple of years ago now, a piece of software called Predictor, which is a Shiny app. You can see a link down in the bottom left where you can go and use the current version. And basically that's a piece of software that comes before you get to your search in a systematic review, and it tells you, it's a way of estimating based on data how long your systematic review might take. We're pretty happy with that piece of software. We wanted to make it more available to more people and to fit it in as part of Metaverse, so we're developing that into a package version. So that'll do everything that the current version does, the online version, but we're also hoping that it will be able to bring in more data to support the calculations that it makes. So the initial version was based on calculations of screening efficiency, how quickly people could screen data. Some of it was self-reported, some of it was from surveys, some of it was from actual experiments. So as robust as we could make it at the time, there are now screening tools like Sysrev that are willing to share some data on how quickly people do their screening. It was based on a much larger sampling pool. So it's possible to make those predictions better through time, and we're hoping to link with Sysrev to do that in the coming months. At the other end of the systematic review process, when you get to the point where you may wish to present not just a report, but also data that you've collected from a synthesis process, we already have the EviAtlas package, the EviAtlas app. So I didn't develop this. This was by a team during our first hackathon in Stockholm in 2018, who put together a general purpose tool where you could drop in your own data and slice and dice it and visualize it in a range of ways. Very useful piece of software for displaying the results of systematic reviews or systematic maps. And basically we're hoping to just wrap that in a package and distribute it. The benefit of that is not just that you'll be able to use those visualization tools without an internet connection, but also that you'd be able to generate your own Shiny apps that display data from your own review, and launch that, so that you could, without having to put a lot of time, effort, money into developing interactive software of your own, you could supply a data set, write a couple of simple lines of code, maybe some logos or customization of the design if you really want to, and be able to put that online very quickly. So it takes the existing functionality of EviAtlas and makes it more broadly useful. So what we're not, there are a range of things that we're probably not going to do though as well. So R it was built as a piece of statistical software. Clearly it has more uses than that now, but it also hasn't developed to the point where you can do, it's sensible to build things exclusively in that. And I think that reflects partly as well the systematic review technology landscape, in that there's a lot of software out there already, and there's no point us duplicating some of that thing. So an example of that is article screening. There's a lot of online tools for doing that. In Metaverse, we have RevTools, which enables you to do some basic screening and particularly some interesting visualization that can help you with your screening. But we probably won't push that to the sort of stage where we're doing things like dynamic recommendation of articles, simply because those tools are already well supported elsewhere. The other thing that we are probably not going to do is duplicate functionality that's already widely used in R, but outside of Metaverse. So there's a package called Bibliometrics, which does some of the same things as the Metaverse packages do. It can import data, it can give you some basic plots of it, but it is particularly well focused on citation networks, and we're not aiming to duplicate that. In fact, we may, I'd be really interested to talk to the team working on that and see if there's some ways of increasing our integration with them, though I don't necessarily know that they want to be part of Metaverse, but we could, that's not a hindrance. So those are things that we're probably going to avoid, despite being potentially useful parts of the systematic review process. So I'm going to leave it there. I think I've hit my 12 minutes or thereabouts. I do want to point out that while I have worked on each part of the work that we're talking about, I didn't lead a vast majority of it, and this is all work that has come out, well, all of it. Large parts of this have come out of our hackathons, and so are led by teams of people that attended them. And particularly for Metaverse, there's a team there that I think you should check out if you're interested in seeing a bunch of people who really did the work. That said, it's a collaborative project. Happy to answer any questions people might have on via email or Twitter or through the chat afterwards. And yeah, I hope you find that helpful. Thanks for your time. See you later.
Thanks so much, Martin. We've got a couple of minutes left for questions, if anybody has any. And we have one question that's already come in that asks, many researchers only do a systematic review once in their project. Do you see them learning R especially for this, or do you see a role for central academic support services to aid researchers in this part, for example, information specialists? Martin, do you want to answer that one? I've popped an answer in there already, but it'd be great to hear your perspective.
Yeah, it's an interesting point, isn't it? And this is something that's come up. Indeed, RevTools as a screening tool was included in a software review, and it got down-weighted because you do need to know code to use it, and that is going to be a hindrance to people. Look, I work in a university, and there we have a bit of freedom to install things like on our machines, but it's worth bearing in mind that some people won't even be able to get to that point where there are workplaces where the use of software like this is even restricted. So even if people wanted to, it wouldn't. I guess I'd say that we've built this software to be as user-friendly as we physically can within the constraints of the of using a coding language. We've written as much documentation as we can. We've put up websites that are as non-technical as possible. We've made so the way you interact with R is by writing functions that perform transformations on data. We try to make those as intuitive as possible and document them as well as we can. So I think to the fullest extent possible, we've lowered barriers. Yeah, I think the counterpoint to the fact that it's difficult, because it is, and it won't be suitable for everyone, but because it's free and open source and fast moving, there are things you can do in R that you just can't do elsewhere. One of them, a non-trivial one, is you can email me or any of this team and say, hey, I want to be able to do this thing, can you? And typically the answer is yes. So, or it's, we could do it with an hour's programming time, or here's someone else who can do it for you. So there are tools that exist well, not just only in R, but you could make the same argument for other programming languages like Python. There are advantages to using R as well as disadvantages. So I guess everyone's gonna have to weigh that for themselves.
Great, thanks so much, Martin. So we'll move straight on now to our next presenter, Christian Cole. Christian is a senior scientist at the Julius Kühn Institute here in Germany, where I am, and he is responsible for the Kadima review management tool. Over to you, Christian.
Yeah, thank you very much for this introduction, Neil. So I hope that you can all see the screen, and I welcome you all to my talk on a tool that we developed here at JKI, which is called Kadima. So Kadima, what it is, it's a free web tool which is supporting the reporting and also the conduct of systematic reviews, systematic maps, as well as further reviews, and very importantly, it is permanently maintained and also further developed here at JKI. So as I said, Kadima covers the entire review process. So it supports the literature search stage, which includes the identification and also the removal of duplicates. It supports the study selection process, whereas currently the same selection criteria have to be applied at title, abstract, and full text stage. But we are currently working on an update, which we are planning to release beginning of November 2020 at latest. And this update will allow users to apply different selection criteria at title abstract when compared to the full text stage. It will allow you to perform a consistency check also at full text stage, so currently only title abstract stage is possible. And it will also allow for the highlighting of keywords at title abstract and full text stage. Besides to this, Kadima also supports the data extraction and also, if you want to do this, the critical appraisal. And very importantly, it provides a very thorough documentation of the entire review process. And if you want to work with the tool, you just have to go to the webpage of it. So it's www.kadima.info. And we're very happy that Kadima is being used. So up to date, we have 2,250 registered users, and we have 2.7 million uploaded risk entries and 47,000 uploaded PDFs that people review teams are working with. In the following, what I want to do is that I want to very briefly dig into different aspects of Kadima, not all, but I think the most important. So starting with the literature search, so very importantly, Kadima is not a meta search engine like Web of Science, PubMed, etc. But what you have to do is that you can exchange reference lists, for example, with EndNote or any other reference management software as a RIS file. As I said, so Kadima does support the identification of duplicated records, and it takes Kadima around 3 seconds to screen 30,000 uploaded records. And very importantly, new entries can be uploaded at any time throughout the review process, and you can rerun the duplicate identification script, etc. So what Kadima is doing then in the next step is that it combines the different bits and pieces you uploaded here and merges them in one list, which serves then as the pool of records that will move on to the selection process. And what you can do at this stage is that potential duplicates that were identified by the system can now be removed either manually or by using the automated duplicate removal function implemented in Kadima. And just to provide you with some numbers, so the clearance of 30,000 duplicates takes around 3 minutes. And the aim of this step is that you get rid, let's say, of all the records that are highlighted in orange, which are actually records where Kadima thinks that there are duplicated records where some validation is needed, either manually by you or by using the automated removal function. Very importantly, as Kadima just adds the different lists you upload to the system and provides this merge list, what you can do here is also you can randomize this list in order to prevent that you introduce a bias just by the order you uploaded the references to Kadima. So now we are coming to the selection process and how to set it up. So in the first instance, you can decide if you want to apply the criteria first to title, second to abstract, then to full text, or if you want to apply them to title abstracts together, followed by the full text. And also you can decide at this stage, you can decide on the amount of records that need to receive an independent and parallel assessment. And this you can do for title, abstracts, and full text. When you did so, the next thing is that you can actually start or applying the selection criteria. For sure, you can also do a consistency check, but I do not show it now in this frame due to time constraints. So with regard to the application process, so there are no restrictions with regard to the number of participating reviewers. So we had review teams where 12 people were engaged when doing the selection step, and this was running very smoothly. And how Kadima allocates this is that it automatically allocates records to participating reviewers, depending first of all on the amount of predefined records that need to be rated independently and in parallel, so what you did in an earlier stage. And then it considers the time point when the reviewers proceed through the progress. And how it looks like, so for example, now at title abstract stage, so you always have a progress bar, which depicts, first of all, the overall progress of the review team depicted in blue. Then you have the progress with regard to the parallel assessment, which is depicted in orange. And then in green, you have your contribution to the overall progress. And how it now looks like specifically at title abstract stage, so it's quite straightforward. So on the right-hand side, you have the title, the abstract. On the left-hand side, you have your criteria. So these pictures are taken from the developer version of Kadima, which already implemented this new update. And we are currently testing it. And as I said, it will be released beginning of November at latest. And what you can also do is you can always attach comments to specific criteria, and these comments will then be presented when you download actually the documentation at the very end of the progress. Also, what Kadima is doing is that it realized in the case of an independent and parallel assessment, when inconsistencies do occur in the rating decision, it highlights it and asks then the reviewers to clarify the inconsistencies. And how it, for example, looks like at title abstract stage. So if you then click on a record that is presented by Kadima, you can see your ratings, you can see the ratings of your review team member, you can cross-check the abstract, and then if you see that you are wrong, you can change your decision. And in order to track the clarification of inconsistencies, you can then just place a comment or attach a comment to the criteria that you actually changed, and like this, you can track it. And at the very end, as I said, when downloading the results, the comments will be included as well. And then you can, actually track it very easily. Now we're coming to the full text screening stage. Similar to title abstract screening, on the right-hand side, you now have your PDF. You have the keywords being highlighted as we predetermined at an earlier stage in different colors. On the left-hand side, you have your selection criteria. At the full text stage, Kadima allows for a bulk upload of PDFs, and in addition, records can be excluded based on reasons which are not connected to the selection criteria. For example, if a full text is not available, not accessible, if we're dealing with a misduplicate, or if it's just a review article without presenting primary data. And also here, similar to the title abstract screening stage, discrepancies will be highlighted, and the review team is asked to solve them. Furthermore, you can also change the view. If you think that this PDF is a bit too small, you can also have a full size version of it, and while scrolling through it, you can have a look at your criteria, rate the criteria, and close the window again. Now coming to the next stage, which would be the data extraction. So what Kadima does allow is, so it does allow review teams to customize their data extraction sheets. And afterwards, Kadima allows for an on and offline data extraction. So in case of an offline data extraction, you can just download your data extraction sheet as an Excel file. You can then fill it in on your desktop and upload it back to the system. Or you can also do it directly online just by clicking on the studies that are presented here, and then a modal is opening, and you can just extract the data, and you can also use predefined code sets in order to streamline the data extraction for certain variables. Very importantly, you can also tell Kadima which variables are relevant for the critical appraisal, and they will appear here in orange. And only those variables that are marked in orange will then be displayed during the critical appraisal. In order to prevent that, we have to go through the entire data extraction sheet. Kadima just presents you those variables, and then it's more easy, let's say, to do the critical appraisal. Yeah, so I think now I'm coming to almost the last slide, and it's now related to the documentation, what Kadima does provide. So what Kadima is providing, it's providing a flow diagram depicting the selection process, but it provides you with the results of the consistency check as an Excel file. We have a file which is called outcome study selection, but what is included here are the records that are excluded at the different screening stages, together with the reasons why they were excluded, and also with comments that you might have attached to them. You get your references or records included after title abstract screening, both the Excel and RIS files. You get the reference included after full text screening, also both Excel and RIS files. You can actually also download the PDFs that you uploaded to Kadima, or only download the PDFs that were included in your review. you get the compiled data extraction sheet, and also you get so the outcomes of the critical appraisal both as an Excel file. So last but not least, in order to be updated on Kadima, you can follow us on Twitter. We are very much depending on user feedback, and this feedback, or in order to see how Kadima is working, you can either test it directly or you can register to online workshops that we frequently do provide. And based on the user feedback that we get, we prioritize the next steps actually with regard to the further development of the tool. And just to introduce the team behind Kadima, so we are not plenty, we're just two. So on the one hand side is Stefan, and he's responsible for the programming work. And it's myself who's responsible for, let's say, the scientific input. And so I'm at the end of my presentation, and I thank you all for listening.
Brilliant. Thank you so much, Christian. So I'm not sure we have time for questions right now, but there is a question that I passed over to you, Christian, if you could answer it in the chat box that pops up. And the question asks, can you choose parameters for when to consider two articles duplicates? So watch out for that. Christian will answer that in the questions window. We'll hand over now. Ooh, sorry, Christian, go ahead.
Where is it?
It's in the questions window. I passed it over to you. If you can't see it, I'll pop it in the chat as well.
Okay.
We'll hand over now to Alexandra Bannock-Brown. Alexandra is a postdoc in meta-research at the Berlin Institute of Health. I hope I got that right. Thanks very much, Alexandra.
Hi, everyone. I hope you can hear me. I'm going to be talking today about a project called the Integrated Interface, which is a project I helped start on based at the Institute of Evidence-Based Healthcare at Bond University, and something that we worked on also at the Evidence Synthesis Hackathon last year in Canberra. So this is really a kind of emerging framework type project still in development that we're hoping can help realize some of the goals of the international collaboration for the automation of systematic reviews, namely integrating the evidence synthesis tools together so that we can more easily facilitate their use. So there's over 78 million records in Web of Science, and if you are trying to keep up to date with a topic, for example, coral on Web of Science, in 2019 there were over 3,300 articles published on coral in Web of Science, and that equates to 9 articles that you would need to read per day in order to keep up to date on this evidence. Now imagine you're a grant reviewer in biomedical sciences. You'd need to read a total of over 1 million articles per year, which equates to about 153 articles per hour. So systematic reviews, I don't need to preach to this crowd, but are of increasing importance. But as Neil touched on, the data that we are trying to synthesize is growing, and it's increasingly complex. So these challenges we're trying to overcome by developing tools and software to help us along each of the steps of a systematic review or a systematic evidence map. And yeah, these can include anything from APIs to link into databases to help make your systematic search living, machine learning tools to aid your citation screening, text mining tools to annotate and group articles or extract information. And for those, you may use any variety of tools along the right-hand side for one or several of these stages. However, there are a number of barriers to using some of these tools, as Neil touched on. There can be a steep learning curve. Tools may be accompanied by a varying amount of documentation, from little to none, to being incredibly user-friendly with many instruction guides and videos, etc. And using a tool for your systematic review may also be dependent on your programming expertise that you have within the team. There can be a lack of support for using the tool. It may not be clear when it's appropriate to use the tool. And yeah, there can be a mismatched workflow. So if you want to use one tool for one stage of the review and move on to use a different tool for a further stage, then these might not be compatible. So these barriers actually don't facilitate the aim of automation tools, which is to reduce human effort required to produce our knowledge syntheses. So utilizing several tools can actually increase the workflow, which is not our aim. Another key concept in systematic reviews, as well as the primary data that we synthesize, is the FAIR principles. In this case, I'm going to touch on interoperability. So for automation tools, we aim that tools are interconnected to allow for a seamless exchange of data. And we want people to have the choice to be able to create a customizable pipeline for their review. Systematic reviews are becoming more complex, but also the range and the types of questions, research questions that we're answering are also growing. And as also facilitated by the evidence synthesis hackathon, new tools contributing to a shared and interconnected system so that we can actually bring them together when one tool is no longer able to be sustained, then we can either there's a similar tool to replace, or we can provide documentation and support to further that development. So that's something that we were aiming to get closer with the integrated interface project. We aimed to develop some technical standards that improve the interoperability and improve the documentation with the aim of increasing transparency as well as accessibility. If tools are documented well, then they hopefully are also able to be found and used by the researchers. And to build an integration interface, a platform or a system that actually integrates these tools that provide this minimal set of documentation into a seamless workflow in order to reduce the barriers and the workload associated with using several tools in an automation pipeline. So I'll just briefly cover what we mean by technical standards or documentation, just the minimum information that's required to use the automation tool and to understand what it is that it does. So we can provide information about the input, what data format does the tool accept, information about the process, what happens to the data when you put your data into the tool, and output, what format is exported from the tool, and how do you receive your data after the process is completed, as well as additional technical information such as licensing and versioning, settings, and container information. And this is all documented on a GitHub repository as well as a preprint. And I just want to take you through an example that we often see in a systematic review process as to why we wanted to develop an automated workflow. So you might have a living search in PubMed, which is which you use via an API, and this returns data in a JSON file format. However, it's a systematic review. You want to search several databases. You also search Embase manually and receive an XML file. You then want to deduplicate these results using first EndNote and then the Systematic Review Accelerator tool. Then convert these files, export them from EndNote in a RIS file in order to use robot search, which is a machine learning assisted screening tool to identify RCTs. And then you might want to get those files out and then port them into an online platform such as SURF to conduct manual screening. So along this process, although you're using automation tools to help your systematic review, there are a number of manual steps, and converting files from one format to another can also increase errors. So that's why we wanted to also develop a interface which would link individual systematic review tools that are also written in different coding languages so that you could use them in one platform. So next, I just want to share with you some of the progress that we've made in creating this overview and implementing some of the technical standards that we've set out. The idea is that you can use the interface either for a single task, like you might use one tool for a single task, as well as linking tools together so that you can use several tools in a single pipeline without having to take your data in and out of separate tools. You can also use, I guess, the apps overview like a kind of app store, plug and play the apps that you want to use in your review, and the data from one step of your review to another review will be automatically sent to the next process. So you can also use it as a kind of Dropbox for reviews. So although this is an emerging framework, it's still in development. We're working towards being able to reduce the barriers for using tools for effective research synthesis. So not only these tools being available, but increasing documentation so they're accessible to all, and they can also be used within a wide range of case scenarios, as well as increasing the interoperability, both to ensure that our systematic reviews are as transparent and as reproducible as possible. This can, with the ease of exchange of data, we hope that these processes can be more transparent. And using these kinds of tools, we hope also as part of the ICESA guidelines and the Vienna principles to have an effective ecosystem for evidence synthesis to reduce any barriers between researchers utilizing tools, programmers developing tools, and aim that there can be a continual improvement so that the needs of researchers utilizing tools, documentation, etc., is provided as and when, so that we hopefully can use these tools to the best of our ability and synthesize the evidence that we need in order to be able to make evidence-based decisions about our science. So I think that's everything from me today. Please feel free to get in touch or tweet me if you have any questions. Thank you very much.
Thanks so much, Alex. That was really, really interesting. Thank you. We've had one question come in while you were speaking, which I've pinged over to you. If you could answer that while the next person is presenting, I'll also send it in the chat, which asks, could you define what you mean by living search and how it's different from what we already do from Farhad Shokranam. And we'll move straight on now for our next presentation by Michael Gusenbauer. Michael is a senior research fellow at the Institute of Innovation Management at Johannes Kepler University, Linz. Michael, over to you.
Okay, thank you, Neil, for the introduction. Actually, I'm working currently at the University of Innsbruck, so Johannes Kepler University was my previous affiliation. So what I'm talking today is evidence identification and systematic reviews, pitfalls and a way ahead. And I'm really interested in evidence identification for a long time. So basically, it started with me being interested in searching. So I used to like searching for furniture, for online movies and stuff like that. And now I'm interested in evidence. In my field is management, but I guess the methods we apply and use for identifying this evidence are interdisciplinary, and I'm very glad I can be here and talk about this. So here I have a picture, a big wave versus my workday, which is basically the same argument as the last presenter. So Alexandra actually made the same point. I have a different metaphor for the same thing. So we are kind of chronically information overloaded, and this now has become very evident in the COVID-19 crisis. And this is the picture from Science magazine that kind of show how medical workers, policymakers, the public, and we researchers are actually overloaded in numbers. So basically, you see I have the same approach here. So we have a lot of papers to read every day. So medical workers, it's about 400 papers a day published on COVID-19. But if you're an electrical engineer, it's 400 papers. Environmental scientists, as most of you are, it's 250 papers a day. Radiologists, it's slightly less, and in my field, it's 72. So it's a lot of papers to read, and this is not getting any easier, as you see here. So actually, 2005, it was, and this is a Web of Science core collection data. So the 10 most, the 10 indices that are included in there in my system. So it's, as you have heard, it's 78 million in total, but what's interesting here is the trend, and the trend is, it might look like an exponential curve, showing you that 2005, it was 1.5 million records a year, and now it's more than 3. So it actually doubled within the last 15 years. So it's a problem, and if you look at Google Scholar, we even have more publications to consider. So it's a factor 3 or 5 times the articles you find on Web of Science, you find on Google Scholar, so considerably larger coverage. So we need systematic literature reviews. I guess we all agree there, but what we also need is, and that's not only including us researchers interested in lit reviews, we need to find better. So it's, even though you might not work on meta-analysis, you still struggle with this information overload. And that's the intro to what me and my co-author Neil are interested in, is the search systems, which are the gatekeepers that kind of between this knowledge we produce as a collective and us reusing that knowledge, and also the public we aim at informing. So we have a lot of different search systems. This is a selection of 28 popular ones we described and analyzed in our paper in research synthesis methods, and we analyzed those with 27 test criteria. And we actually found out that, as you know, systematic reviews have unique requirements. So you want to identify all records within your resource constraints, obviously. So it takes a lot of time, but you also need to be reproducible and transparent in what you're doing. And if you adhere to these criteria, you cannot use all of those systems. So maybe you can now think and pick the ones you are regularly using and think about some advantages and limitations they have. And that's basically what we did in the paper. And we found out that actually only half of them are liable to be used as principal systems, which means that they have specific capabilities that are typically needed in systematic reviews, like Boolean searching. So the building blocks heuristic you are all familiar with, and we also looked at search string length and a host of other things. You can look it up in our paper. And so we found out that only half of them can actually be used as principal systems. Principal means the main system you're using, of course, with different heuristics like citation searching that you do additionally to the main search, which is typically a Boolean search string. You can do on those systems, but that's just supplementary to the principal system you use. So that was actually already revealing that we have to be careful in selecting which system to use. But now we have an issue because this landscape is actually changing, and you ask yourselves, well, that's 28 systems, but I use a different one which is not included here. So what about this one? Or also about like what's about dimensions.ai or lens.org or the newly coming method.org by the Chan Zuckerberg Foundation. So this environment is constantly changing. So the paper publication format is, which is quite static, is not the best for that. So this is why we are going a step further and using the methods we implied in this paper, updating them to some extent. And we are currently working on Search Advisor, which is a web-based tool to compare academic search systems. So the ones you've seen in the previous slide, plus some extra ones with a few clicks. So it should be quite user-friendly, not just for reviewers, but also for any PhD, master students, anyone really who is interested not only in systematic reviews, but just, okay, where do I find the most papers for my discipline, and where I can do a Boolean search, or where I can do whatever. So it's it should be a very straightforward tool to allow to do that. And so there's diverse use cases, and we will use predefined filters, and it's really available. I'm not confident to show you any details at this stage, as it's still in pre-alpha mode, but we received funding from the Austrian Science Fund. So we are hoping to get in beta in spring. What are the benefits? So to researchers and librarians, you can compare, and you have transparency in what works and what doesn't. And in the next slide, I will show you what that means, why this is important. For the search systems themselves, it helps to illustrate what is important for us as systematic searchers for systematic reviews. And it also gives them a benchmark. So they can see, okay, how are we performing compared to other systems, to encourage further development. And I have an interesting example here, which illustrates the necessity of these comparisons and the increased transparency in the search system landscape. And this is an example of Semantic Scholar. Maybe you are familiar with that search system, using artificial intelligence to curate records. And this is 3 screenshots over the course of 1.5 years. And we actually identified in our paper that a central functionality is not working with that system. So the Boolean search is not working. And even though they reported that they would support it in 2019. So it was actually to some extent incorrect. And after we published our paper, and it might be a coincidence, I don't know, but it's after we actually found out that this system is having flaws in Boolean search, they kind of changed the FAQs to now it reads they don't support Boolean operators. And this is an example of how we really have to be careful so we really need to question what the functionalities are that are offered by these systems, and if they really help us to find the stuff we want. And to wrap up here, so what's next? I hope it's okay to use this format to spread a questionnaire, which is basically about the criteria that you find important or not so important of search systems, which helps us as the search advisor team to further develop the software. So it would be nice if you could answer that questionnaire. And Jessica, I see she has already posted the link. It takes 10 minutes, and it would help us to know what is important to you as systematic reviewers to help improve our system. So thank you very much, and I hand over.
That's great. Thank you so much, Michael. That was really fascinating. We don't have time for questions just yet, so if you have any questions for Michael, please pop them in the questions box. We'll now pass on to Eliza Grames. Eliza is a PhD candidate at the University of Connecticut, and she is the author and developer of the popular searching tool in our Lit Searcher. Over to you, Eliza.
Okay, thanks, Neil. So yeah, I'm talking about ways that we can increase efficiency of searching, which is kind of the goal of a lot of us here, is just how can we make this more efficient. And so, like Neil and Michael and Alex, I wanted to point out that the number of publications has been increasing dramatically over time. And it doesn't take much to imagine that the cumulative number of publications has also increased a lot, which means that there's a lot of evidence to synthesize, which is great for those of us who work in evidence synthesis, but it's also really time consuming. So there's this predictor tool that Martin had mentioned, which Neil and Martin developed, which helps you estimate how long it will take to do a systematic review or a systematic map. And the average time is 328 days for a systematic review. And you can imagine this is only going to increase with the number of publications out there, because a lot of time is spent on title screening, abstract screening, and then all the steps after that. And so I think that really focusing on searching is really important, because it's kind of foundational for all subsequent steps. If anyone knows a way to automatically formulate a research problem, then I guess we'll all be out of a job. But once you get to finding and selecting studies, that's where you can really start automating this process, which will help build the foundation for everything that comes after that. So it doesn't matter if you are, doing the best work you can, coding studies, calculating effect sizes, if you haven't actually found all of the available evidence for doing a meta-analysis. So I think it's a really important step, both in terms of time and also how it can affect the end conclusions of a systematic review or meta-analysis. And so to have a good search and to be able to be confident that you've actually found the information, but you're not spending too much time, you need to be able to balance the precision and recall of a search. And so this gray box here represents the idea of like all of the possible articles. So everything in Web of Science, Google Scholar, and out there that's published. And in this example, we somehow know what all of the relevant articles actually are, and so that's this pink box. And so ideally, our search should be able to retrieve that entire box of articles without retrieving anything in the gray box that's not relevant. And so to try to capture that, of course, the world's not perfect, so we can't actually get just that box. We could either do like a very high recall search, so we've retrieved a good portion of the relevant articles, but the search also has really low precision because we're also screening all of these irrelevant articles, which is going to increase the amount of time that we have to spend on the review. We could also do a search with fairly low recall, so we're only getting, maybe a quarter of the relevant articles. that's really high precision because we're only screening some irrelevant articles. So we're not going to spend a lot of time looking at articles that aren't relevant to the topic of the review. But of course, we're missing a lot of evidence. And so trying to come up with a way to actually balance your search terms to get that exact sweet spot is really challenging. So how do we actually choose the best search terms? So a lot of people would probably say, use like a standard dictionary or an ontology like medical subject headers, which predefines what types of terms might be associated with the evidence you're looking for. But for my fields, for ecology and conservation biology and many other fields, there isn't actually an ontology like medical subject headings. So we don't actually have a standard set of terms. So for example, I pulled these screenshots from ResearchGate of people asking to define terms. Can anyone help with this terminology, process versus mechanism, function versus role? What's the difference between the terms cosmopolitan and ubiquitous? And then are cluster roots and proteoid roots interchangeable because some authors switch between them in different publications, which suggests that if you're looking for articles on cluster roots, you might also want to search for proteoid roots. I don't know if they're actually technically different things, but it suggests that somewhere in the literature they've gotten confused, so you'd want to use both of them. But how do you actually come up with those terms if you don't know in advance? And so the way that a lot of people do this is just relying on subject expert knowledge. So for example, this is a systematic review out of the lab that I'm in from about a decade ago. And they did a really high precision search, but with not necessarily high recall. So we came up with this list of search terms. So they heard the terms area sensitive, fragmentation, and patch size with bird and passerine to try to find articles on birds in forest fragments. And if you run this search in Web of Science, you get about 4,000 results. But if you add just a couple synonyms and modify it a little bit to include like area sensitivity and patch size or forest size, you get more than double the number of search results. And so it's really critical to think about like what evidence are we missing by not including all of those synonyms. And so that's the background for why I developed LitSearcher, which is an R package to help with automatically identifying search terms for systematic reviews. And so I just want to go over the logic behind LitSearcher and how it works, because that's one of the things that's most important to understand about any kind of tool that you're using. So how LitSearcher works is it expects you to gather a set of relevant articles. So you come up with your best faith effort to do a search to capture articles related to the topic of your review, and then you feed those into LitSearcher. And so first you have to import the results, and actually these functions have been moved to the synthesizer package that Martin mentioned to reduce some redundancy. So Martin and I had been independently developing functions for importing and deduplicating search results, and so we merged those into the synthesizer package. And so LitSearcher will call synthesizer to import bibliographic data files and then remove duplicates. And then the next step is to extract potential keywords. And for this, it uses an implementation of the rapid automatic keyword extraction algorithm, which is going to look for nouns, adjectives, and adverbs that are next to each other, that aren't separated by punctuation or stop words. So for example, as and a are stop words, and so they're separating patch openness and causal mechanism into different groups. And so it'll go through, extract all of these potential keywords, and then put them into a keyword co-occurrence network. And so two terms are co-occurring if they're in the same set of title, abstract, and keywords. So for example, patch openness and causal mechanism are co-occurring in this article. And so LitSearcher will build this keyword co-occurrence network, and in the network here, each circle is a node in the network, and it represents one keyword, and then the lines between nodes are co-occurrences. And the idea with putting things into a keyword co-occurrence network is that all of these bigger circles in the middle of the network are the important terms in a field of research. And so those are terms that are good candidates to include in a search strategy, whereas things on the outside here are probably less relevant or not used as frequently. And so LitSearcher will select this subset of important terms and then suggest them to researchers to then go through and manually review and see, does this actually meet any of your concept categories for your search? And then users go through and manually group those. So for example, I went through and I was like, yep, it has found this word for a bird and another bird. So some terms that are not relevant and then some that are synonyms for area sensitive. And so once users have grouped all of those together, then you can pass the whole list of terms back to LitSearcher, and you can add other terms, of course, as well. And it will write a Boolean search for you. There's a bunch of different ways that you can ask it to write it. So for example, here I said that I want exact phrases, so to put things in quotes, and truncation. So it will automatically truncate words and then put the asterisk to stem them, so accepting different forms. It will also translate searches into up to 53 languages by calling Google Translate API from within R. So if you don't have someone on your team who can speak a different language, but you want to search in that language, you can have LitSearcher translate your search for you. And so you might wonder, how does this actually compare to if we did it manually? So I replicated 6 systematic reviews and maps from environmental evidence. And I took the original search from the published review, which is the replicated one, and then I followed the LitSearcher process to see how well they actually did at retrieving the original articles from the review. And LitSearcher is doing pretty good. So not too much worse than the original searches, but way, way quicker. So it took me about less than 2 hours total, whereas in general, it takes like 25 hours or more to develop searches. And I was pretty pleased with this, especially because I had to do this process for a lot of topics I don't know anything about. So I study birds, so like I had to write searches for mesophotic coral reef ecosystems. I also compared how LitSearcher had performed in this test with the results of similar tests for other automated methods. And so this citation networks approach was doing much worse than the replicated searches. So it was a combined method of text mining and citation networks. And then there's this text mining method developed by L.P. Hausner, which also works just as well as conventional methods, but both of them are having kind of low recall issues, which suggests that a lot of those articles were being retrieved from other search methods. And so if you want to learn more about the methods underlying the package, you can read the paper that is in Methods in Ecology and Evolution.
Great. Thanks very much, Eliza. That was really interesting. I definitely recommend people take a look at that package. I think it's really useful. So there is a little bit of time. Max on the panel has a question, actually. Is it possible that recall might in fact be higher, given that LitSearcher may identify relevant articles that were missed in the original paper?
Yeah, that's definitely possible, especially because in I think 2 out of the 6 reviews, the search strategies that I developed with LitSearcher did retrieve articles that were included in the original review that the search for the original review didn't actually retrieve, so that were probably found through like hand searching or that they already knew existed. So we do think that recall is probably higher. We just couldn't actually check that because I didn't go through do the inclusion criteria for all of the articles we got back, and we're just using the results from the original one. But yeah, we do think that recall is probably higher, so it helps with doing a more comprehensive search, but also targeting it into more specific ones.
Great, thanks very much. And then a follow-up question, just very quickly, if you could answer, how did precision compare?
Oh yeah, it was less precise in a lot of cases, and some of those I'm not sure if it's because I didn't know exactly what I was doing for the naive searches, like when I was looking for coral reefs. Yeah, so it's a trade-off. So it was less precise in some cases and more precise in others, kind of depending on the topic of the review. And that's something that like users can change in the settings too if you want more strict keywords or to be more broad. Yeah.
Great, thank you so much, Eliza. We'll move on now to our next speaker. Just before I do, Ko Kono, who's asked a question in the audience, you've asked a question, but I can't see the question. So can you just double check your submission, possibly ask that question again? Meanwhile, we will be moving on to Jan Minks, who is head of the Applied Sustainability Science Working Group at the Mercator Research Institute on Global Commons and Climate Change. and is the Priestley Chair of Climate Change and Public Policy at the University of Leeds. Over to you, Jan.
Thank you so much, Neil. Hello, everybody. I'm Jan. Just as a caveat, there's construction work that just started half an hour ago, right above my office. They are drilling. I don't hope that this will affect us too much. Also, quick thanks to Neil and Martin actually for putting this on. I think it's really fascinating, and I'm really genuinely enjoying it. Maybe before starting, just to give you a bit of a perspective where I'm coming from, I'm more kind of I have been moving into the field of systematic reviews, but I'm coming more from kind of global environmental assessments, like as those conducted for the IPCC. And I wanted to start with that, but I can't actually move my, I know, here. To also show one of those, but with a focus on the IPCC. So the different colors of the bars is the literature that emerges during assessment periods, so assessment cycles where the IPCC conducts its reports. And the IPCC as an assessment body has a kind of comprehensive mandate to actually provide like a comprehensive assessment of the entire literature. And if you just look in the Web of Science, we have on climate change quite narrowly searched for more than 45,000 publications only in the Web of Science. That is roughly the literature that we had for the first 3 entire IPCC assessments. And now for the AR6 that is currently running and where I'm contributing to, we expect about 350,000 publications, again, only in the Web of Science. If you look at the entire climate change literature, including gray sources, we think that there's definitely, consistent with the query that we used, about 1 million studies out there right now. And so that's really our perspective. So the IPCC at the moment has no ways of tracking that literature. And so we are coming really from this idea that you actually want to use machine learning to actually look at entire research fields as well. And only later on we also got more into the specific systematic review processes. And this talk is really about that. That I want to show actually different perspectives on evidence mapping that you can have, and how machine learning may contribute. And it will give you a few impressions of kind of capabilities that we have been building here in Berlin over the last few years. So to start with, I thought it's kind of useful to say, okay, what types of machine learning do we have? So and I'm mainly focusing now on the machine-based coding extraction of data. So we have supervised methods like vector machines, like neural networks, etc. We have also unsupervised machine learning methods like topic modeling that we can use, and then we have kind of semi-supervised methods like keyword-assisted topic models or so. And within those unsupervised and semi-supervised methods, there are also other variants like dynamic versions of topic models, for example, or structural topic models, where covariates, where metadata is used to actually to control for the influence of different covariates on topic distributions, for example. And the question for me was, how can we actually how can we use those, or how we can make sense of those capabilities that we may want to learn? And it seems to me that one was, if you want to distinguish, what should I actually do, which capabilities, which tools do I want to use, then it makes sense to distinguish between the scope and the specificity of information that I want to extract. So unsupervised methods, of course, don't give you a lot of control over what you can extract, while supervised methods, like similar to hand-based coding, can actually do practically the same job. The other thing that is really seems important to me for deciding is, what is actually the domain knowledge that you have, and with supervised methods where you code specific things, it's a field. These are applications where you require probably rather a lot of domain knowledge, while there are methods that lend themselves to applications, to evidence mapping applications where you actually don't have a lot of domain knowledge. And there are further distinctions. We can even think about, what level of information do I actually want to use? And I think there is actually a case that for certain evidence maps, you are better off using abstract-based information rather than full text, while there are others where it's the other way around. And so in principle, in that spectrum, systematic maps probably rather sit to the right, and research fields and what the type of evidence maps required by global environmental assessments probably sit rather to the left, even though I will show also really large scale supervised machine learning applications here. So yeah, as a caveat, I'm not considering all intervention points. Just this is a really incomplete effort in just the trying to structure that discussion on machine learning a little bit. So I think this, if we actually want to identify, classify, and map out potentially vast literature with regard to specific criteria, then it's probably something where we use supervised machine learning classification, and I would call this maybe targeted evidence mapping. Here you can see an example where we try to classify the entire literature on climate impacts, and about 70,000 studies it turned out and located automatically, etc. So we can ask quite specific questions, very similar as we do with systematic maps, what the literature is on different climate impacts for what locations, etc. Then there is a completely separate avenue for evidence mapping where we actually have vast fields we don't know much about. And I think that's actually an application for unsupervised methods where we want to gain oversight of a vast and unknown knowledge domain, identify hot and cold topics, overlay with publication-specific metadata probably. And the method here is unsupervised machine learning. We don't actually, we can have blind spots because of the methods we use. And so here we have a real opportunity for applications where we can really sketch out huge, large research fields like the entirety of climate change, as we did here in this nature climate change piece this year. But then I think systematic maps, for my case, are often or tend to be a bit descriptive. And so if we now think of different classes of evidence maps, we can also think about inquisitive evidence mapping. So explorative before and targeted now, inquisitive. And so we want to actually answer a specific research question on the evidence landscape by assessing the influence of different covariates, for example, on certain research topics. The method here would be structural topic modeling, where you can think about this as a combination of a regression analysis on a topic model. And an example would be where we would ask how study and author location, for example, affect the thematic focus of research. So we can actually quite rigorously assess, a set of research questions that we have then on the research landscape. And then finally, maybe also something like layered evidence map. And this is actually now not a specific machine learning application. It can be combined with different things, but it turns out that it can be really, really helpful to overlay lots of additional primary and or secondary data. And here we did this for the climate impacts, where in the field of climate change, it's really important. There are lots of controversy on impacts detection and attribution, and it's really interesting to see which studies that are out there of these 70,000 are actually in locations where the climate impacts can be formally attributed to climate change and not to natural variability. And that actually, in terms of detecting evidence gaps and generating other insights, is really, really interesting. That's all that I wanted to say today. So I think machine learning, beyond going through those various stages and supporting our standard process, also provides opportunities for actually new types of evidence maps. And I think we need to think about that as well. And there is a need to structure this field. Here I proposed a very simple and naive typology for evidence mapping that hopefully can trigger some discussion. Thanks so much.
Great, thanks so much, Jan. That was really interesting. If anyone has any questions, they can pop them in the question box, and I'll pass those over to Jan to answer in the meantime. We'll move straight on now to Max Callahan, who is also at the Mercator Research Institute on Global Commons and Climate Change. He's a PhD candidate there. Over to you, Max.
Thanks very much. I hope you can see my screen and the presentation. Yeah. Okay, so I'm going to talk about stopping criteria for automated screening in systematic reviews. So,
Max, we can only see your web browser right now.
See, that's the wrong one. Thanks very much. Can you see the right one now?
Yes, in full screen now.
Brilliant. Okay, sorry about that. Yes, so machine learning promises to save us lots of work in various parts of the process of doing systematic review. And one of the prime examples is in doing screening, so separating relevant from irrelevant articles in a population of potentially irrelevant articles. So this follows on from what we heard before, that was about developing a query. So once you have a query, you still need to separate the relevant from the irrelevant documents. And if you screen them one by one at random, so then that's the green line on the right of this figure over here, which means that after seeing all of the papers, then you will have achieved a recall of one. But what how machine learning can help is it can learn from your decisions, from human decisions, what papers are relevant and what papers are irrelevant, and give the human screeners the relevant documents first. So this is what happens with this red line here, that after only seeing a fraction of the total number of documents, you've already seen 100% of the relevant ones, so you have a recall of one. The trouble is, we don't know when we've actually achieved 100% recall, so we need to figure out what's a good time to stop and how we can report this. So just to explain the workflow in a little bit more detail, what we usually do is we screen a random set of documents, train a machine learning model, predict the relevance, and to screen the most relevant documents. Then if we think we've achieved the right recall, then we stop, and if not, then we go through another iteration where we retrain the model, predict the relevance of the unseen documents, and screen again. And the trouble at the moment is that we don't have a very good way to estimate whether we've achieved a recall target or not. There are some examples in the literature. This has already been identified as a gap, but there are some approaches that kind of go toward this, and I'll discuss them very quickly. So the first is to take a sample of all of the documents and to estimate from that how many relevant documents there are. And then you can keep screening until you've seen 95% of that number. And if you take a set of 20,000 documents, of which 500 are relevant, and you take a sample of 1,000, then this is the distribution of guesses you'll make about how many relevant documents there will be. The trouble is if you guess too high, then you won't save any work because you'll never get to the number that you're waiting for. And if you guess too low, then you will achieve a recall less than your target recall, let's say 95%. So this method doesn't account for sampling error, and even in really big sample sizes, so up to 10,000 documents, you're still often achieving no work savings and often getting a recall less than your target. So this is quite a big problem. The second one is to look for several consecutive irrelevant documents, and we have to set this number somehow, but we basically interpret this as if we see lots of irrelevant documents in a row, then we guess that the proportion of relevant documents remaining is low. And this is a useful intuition, but we think that it misses something really quite obvious, which is that a low proportion of a small number has smaller implications for recall than a low proportion of a big number. So if you have lots of unseen documents, even if the number of relevant documents, the proportion of relevant documents in that set is low, you still might miss your recall target. So there are also some other papers that have kind of gone a bit further than these and don't have quite the same fundamental problems as what we've discussed, but actually none of them properly account for uncertainty, and none of them reliably meet recall targets, and none of them offer users the ability to communicate when they stopped. And we think that for live systematic reviews, we need criteria that can communicate what are the implications of stopping early in a way that's really clear for non-technical readers, and is also independent of the machine learning approach and the performance of the machine learning algorithm. So I'm going to go over this very quickly, but feel free to ask me questions about the more technical detail. What we do is we start a random sample at an arbitrary point during the screening process, and we form a null hypothesis that we've missed our target, so that the recall is less than the target recall. And what we're doing when we're screening is we're sampling without replacement, which means that k, small k, which is the number of relevant documents in a sample of size n, small n, from a population of n documents with k relevant documents is drawn from a hypergeometric distribution. So we can introduce a hypothetical value for k, which we call k target, which is the lowest value of k, which is compatible with our null hypothesis. And by estimating the cumulative probability of observing what we observed, given k target, then we can see the maximum probability of observing what we've observed if we had missed our target. So if that's below 1 minus alpha, then we can reject the null hypothesis and stop screening. One problem with this is that we don't know when to start this random sample. So we develop a sort of version of this criterion, which we call ranked quasi-sampling. And all it does is it treats previously screened documents as if they were random samples. So this method is actually always conservative, as long as the proportion of relevant documents given to the screener by the machine learning algorithm is greater than or equal to the proportion in the remaining documents. So if the machine learning algorithm is working better than random, then this should be conservative. So we test this as an independent criteria, but we also use it to decide when to stop screening, machine learning ranked screening, and to begin a random sample. So we evaluate this on a large set of already completed systematic reviews, which we draw from various papers that try to develop their own methods for machine learning for systematic review screening. And we do 100 simulated reviews on each of these data sets. Each time we begin with a sample and then draw 10 documents, retrain a model, predict the relevance of the unseen documents, and reveal the real relevance values. So simulate in a simulated way the next 10 documents. We keep doing this until we've screened all the documents, and we record when each criteria would have been achieved. So here are the results that you get if you have a priori knowledge of how many relevant documents there are. So here we're looking at work savings on the x-axis and recall on the y-axis. And of course, you always get recall at or close to 95% because you know exactly when to stop. And you can achieve a kind of range of work savings up to pretty large work savings near to almost all of the data, and also including some not so high work savings where the machine learning hasn't actually helped that much. So we never miss the target, and we can achieve some pretty large work savings. And actually, these are the results that are most often reported in these studies, although they're unachievable in the real world. So this is what happens when we use the baseline sampling, baseline inclusion rate. So when we sample at the beginning and guess how many relevant documents there are. And the problem with this is that sometimes we miss our target for recall, and we often miss our target quite dramatically. So we can achieve recalls close to 0, which is in a systematic review pretty catastrophic. And we also quite often don't save any work. And when we do save work, those work savings are not so useful because we're also missing relevant documents. So heuristics is a similar story. We have a trade-off between work savings and recall, and we often also don't save any work at all. We often also have 0 work savings. If we have a stricter heuristic, so we wait for 200 consecutive irrelevant results, we have better recall, but lower work savings, and we still miss our target quite a lot of the time. So here's our random sampling criteria, where we miss the target in less than 5% of cases, which is pretty much what we wanted because we set our confidence level at 5%. And we achieve relatively modest, but still existing work savings. And here's the ranked quasi-sampling version, which achieves slightly greater work savings and actually is even more conservative. And so we think we can use this, but it's not robust to catastrophic machine learning failure. So if something went really wrong with the machine learning algorithm, then it is possible that you would miss the target more than your confidence level. So to conclude, what we think we provide here are reliable stopping criteria that realize some of the work savings in systematic review screening. We can, previously, the outcome of a badly performing machine learning model could have been low recall levels, which are extremely problematic for systematic reviews. But now the only thing we're risking is work savings, which is obviously annoying to save less work than you wanted, but it's better than missing out on relevant documents. And we think that we can use these in live reviews because we can report the implications of our decision to stop early for recall. So you can say in a live review, we reject the hypothesis, the null hypothesis, that our recall was lower than our target recall of 95% at a significance level of 5%. So one note about the work savings here, they're modest, but we used extremely general machine learning models and did not tune the parameters to perform better on individual data sets. And of course, larger savings are possible in larger data sets, which is exactly where these work savings are most helpful. And in forthcoming work, we want to investigate how work savings depend on data features so we can develop a kind of potential savings calculator for new projects. Okay, thanks very much.
Thanks so much, Max. That was really great. So we'll move over now to our next speaker, who's Wolfgang Bichbauer. Wolfgang uses a Linux system, so he's too technologically advanced for GoToMeeting, which means there'll be a slight delay while we hand over because every time the presentation stops, apparently he gets kicked out. So while we hand over to Wolfgang, I'll just give a brief introduction. Wolfgang is the Associate Professor of Methodology and Statistics at Maastricht University. He's also the developer of Metaphor, a package in R for meta-analysis. Anybody who's done meta-analysis in R will be aware of Wolfgang's work. Hopefully we have Wolfgang back now. Yeah, I can see he's online. We just need to unmute him. Wolfgang, are you able to hear us?
Yes, can you hear me?
Yes, perfectly. Great.
All right. Good. So thanks for the invitation to be part of this webinar. So we've heard quite a number of talks that are about the sort of gathering step of a systematic review, screening and searching. And so my talk now comes really at a later stage where you have already done all of this hard work. And so now you want to do a sort of systematic synthesis of your studies. So you want to do a meta-analysis. And what I want to talk about is the functionality of the R package metaphor, which allows you to create sort of automatically reports based on a particular analysis. So before I get to that, though, I have to say a few words about the metaphor package itself. So let's move to the next slide. All right, so the metaphor package, that's an R package for meta-analysis, and this is something that I developed at the beginning of the century. That seems like a very long time ago. And so this was part of code that I wrote for some research that I did as for my dissertation. I needed to fit meta-analytic models, and so I wrote some R code for that. Then that kept growing into additional functionality. It became a full R package in 2009, and I have released 25 updates ever since. So I keep expanding on the functionality of the package. If you want to read a bit more about the package, there's a paper in the Journal of Statistical Software. And then there's the link to the CRAN site. The package has its own website, and there's a place to read the documentation where it's nicely formatted. All right, moving on. So just to, so the general workflow, once you have collected your studies in a meta-analysis, is that you compute your outcomes, your observed outcomes or effect size estimates, whatever those may be, maybe risk ratios, odds ratios, standardized mean differences. Then you analyze those estimates with some appropriate meta-analytic models, and then you may do some model diagnostics. You usually want to create some figures like a forest or a funnel plot, maybe do some checks for publication bias, and then you need to write this all up for your paper, send it off to Science and Nature, and then become rich and famous, of course. Well, not sure about that last part, but you get the idea. So let's look at a simple example. Next slide, please. So this is on the BCG vaccine, a vaccine against tuberculosis. Was developed around the Second World War. And well, you have to eventually test whether the vaccine is effective by doing human trials. So you have a group of people that get vaccinated, another group does not. You follow them up and you see what is the infection rate in the two groups. So let's look at the results from one of those studies. Next. So here in this study, we see the results at the top, how many people were assigned to the two groups, the number of infections or TB positive cases. So we have an infection rate of about 3.25% in the treated group, the vaccinated group, almost 8% in the control or the not vaccinated group. That gives you a relative risk of infection of 0.41. So almost a 60% lower infection risk in the vaccinated group. Now, for a meta-analysis, we log transform these risk ratios for some statistical reasons. And so we have an estimated log risk ratio of minus 0.89, and then also a variance to go with that. This 0.326 is the variance of our estimate. So that tells us something about the precision of our estimate. Now, we don't just have one study on the BCG vaccine, but there were 13 trials that were conducted on the effectiveness of this vaccine. So here you have the data that are actually also on the metaphor package. So I'm loading the metaphor package and I'm looking at this data set. So T, pos, T neg, that's the treated group and the positive and negative cases. C is the control group, C pos, C neg, the number of positive and negative cases in the control group. There are some other variables that are not relevant here. So let's go through the usual workflow. So step one is to compute the, next slide please, yes, the log risk ratio. So there's a function here, ESCalc, that will do that for you. We specify the effect size measure. RR stands for these log risk ratios. Then we need to give the function the information, these counts for these 2x2 tables. And then I'm also adding some information about the study labels, SLab here. So we will get some nicer figures in a moment. Then the next step is to run a random effects model, for example, on these estimates. So we have these log risk ratios and variances. We use that as input for the RMA function, which is sort of one of the main functions in the package, and it fits a random effects model. So assuming you're sort of familiar with this kind of stuff, we get the usual things you would expect here. We get an estimate of tau squared, the amount of variance in the true log risk ratios. We get I squared as another sort of measure of heterogeneity. We get our test for heterogeneity. We get an estimate, this minus 0.71 here. This is the estimated average log risk ratio, and we get a test for that, a confidence interval. So you get sort of the output that you would hopefully be looking for, but it's all kind of a bit technical, right? So you need to understand what these things mean in essence to really make sense of it. Moving on. You may want to do some further things like create a forest plot, a funnel plot. You may want to do some influence diagnostics, so maybe there are some outliers or influential studies, and maybe a rank or regression test for funnel plot asymmetry. So is the funnel plot symmetric or not? And this may indicate possibly if there's publication bias or not. All right, so that is how you would usually do things. And now I want to talk about the reporter function. So this is something that I started to develop at the Evidence Synthesis Hackathon in Stockholm. And so the idea is that that this function, what it does is it creates an analysis report for you. So it describes the methods that were used in natural language, so it's not quite so technical, or it's more readable, consumable in some sense. You get a forest, a funnel plot, you get also references for all the methods used, and this can be exported as an HTML, PDF, or as a Word document that you can then further edit if you would want to. So let's try this out. I cannot do a live demo, but we just use the reporter function on this object, this model object, and you get some output as the report is being built. And so let's start looking at the next slide where you can see what this will look like. So I'm not going to read everything here, but just the beginning part. So the analysis was carried out using log risk ratios, a random effects model was fitted, and then it tells you how the amount of heterogeneity was estimated. You get these references. Test for heterogeneity, I-squared statistic, prediction interval. It tells you how it checks for outliers, possibly influential studies. Then you get your explanation about the regression and the rank test for potential publication bias. And then you get the results. So it tells you how many studies were included, what was the range of the observed log risk ratios, then pooling them together, you get your average log risk ratio or the estimate, the test, then it points you to the forest plot. Next slide, please. So you get your forest plot, then it tells you about the Q-test for heterogeneity, tells you about the prediction interval, which I'm not going to get into here, but some further information. Let's move on to the next slide. Then in the examination of the studentized residuals reveals that none of the studies had a value greater than particular cutoff that is explained in the methods. So depending on whether there is actually such a study or multiple potential outliers, the text, of course, will change. So this text is dynamically created depending on the results of the analysis. Then you get the funnel plot. Neither the rank correlation nor the regression test indicated any funnel plot asymmetry. So again, depending on the results, it would say maybe one or both of the tests are significant. Moving on. So then you get the some notes here. So how this report was created. It gives a little disclaimer here. This report provides an illustration of how the results of the model can be reported. But obviously, this is not a substitute for really carefully examining the results yourself. But still, this is definitely, I think, a useful start if you want to write up these results and to sort of understand what are important pieces that should be reported. And then you also get references along for all of these methods. So this can save you quite a bit of work. All right, last slide. So that's the idea of the reporter function. It's not complete yet. It cannot deal at the moment with models that include predictors, so meta-regression models, but this is something that I want to work on in the future. Maybe some additional functionality, for example, suppressing the certain plots if you don't need them. The analysis here is done with log risk ratios, but typically we may want to report the results back transformed, so as risk ratios. So you may want to transform the results, and I think that functionality should also be there, that the results are then stated not in terms of log risk ratios, but as risk ratios. Maybe functionality for adding some explanatory footnotes. So the report doesn't tell you what I-square is. It will tell you, it gives a reference to the paper about I-square, and the result is also reported, but it doesn't explain these things. So it may be nice, especially if you want to use this as a teaching tool, to have the option that the report includes explanatory footnotes. So it will actually define certain things and explain what they mean and how to interpret them. Then there are other types of modeling functions for the mental Hensel method, Pedo's method, and other types of models you may want to fit. There are other functions for this, and right now a reporter will not be able to deal with them, but this is just something that just takes time to expand on the functionality of this reporter function to do that. And maybe some more customization. So right now it just says the log risk ratio was this, but it doesn't tell you that this is comparing like a vaccinated versus non-vaccinated group, or that the outcome was infection versus non-infection. So maybe the user could supply sort of text pieces that would then be also added at the appropriate places, although that gets a bit tricky because making this work in all cases, because this is completely general, so this is not specific to log risk ratios. You can use this reporter function on a meta-analysis of correlations or standardized mean differences, and then it gets really tricky to make it this customization really sort of applicable to every sort of scenario. But it's just an idea of something that I may want to consider in the future. All right, so thanks for your attention, and I'm happy to take any questions about reporter or the metaphor package in general. Thanks.
Thanks so much, Wolfgang. That's really, really interesting. Thank you so much. We have one question coming in that I'll ping over to you now, if that's okay. We'll move straight on to our next speaker. Our next speaker is Dale McNally. He is clinical investigator at the Children's Hospital of Eastern Ontario Research Institute, and he's the developer of Insight Scope. Over to you.
Can people hear me?
Yes, that's great.
Okay, thank you so much, Neil. My slides are visible, Neil?
Yes, I believe they are, yeah.
Okay, great. Okay, so first off, so thank you, Neil, for the introduction. My name's Darren McNally. I'm a pediatrician in Ottawa, Canada. I spend half my time doing clinical work and half my time doing research. Big thanks for the invitation to present on a platform that we've been developing over the last couple of years. Just in terms of conflict, because we built this platform and there was code associated with it, we needed to set up a corporation, which is a non-for-profit, and I'm the president of that, so to get that conflict out of the way. So section 1 is a problem. We have 2 problems that we were trying to address with the development of this platform. The first related to systematic reviews, and everyone on this call is very familiar with systematic reviews and their importance and what makes them challenging, and then the downstream consequences of those challenges. So that's a problem everyone here is very familiar with. The second problem that I encountered as a clinician and as faculty was this concept of an unfulfilled crowd, where weekly or sometimes every other week, I will get an email from a student. Sometimes they're a med student, a graduate student, an undergrad, who is really interested in getting involved in research, and they've been struggling and trying to find a supervisor willing to take them on. So as I was doing my own systematic reviews, I was running into this paradox of having a lot of work to do with the systematic reviews and trying to find ways of incorporating students into research, and there being more students than I could possibly help. So we wondered whether one problem could solve the other. When we thought about this, it sounded a little bit like crowdsourcing, this idea of obtaining a needed service by soliciting contributions from a large group of people, rather than from traditional sources, where the traditional source here is your immediate surroundings, what students or people work on in your division, on your same floor, at your same university. So we decided, going back 4 years, to do some research on this idea of crowdsourcing systematic reviews. There wasn't much known at the time, and we've done a number of studies on this, but I'll just present one. We did a validation study, and the gentleman in the lower corner, his name is Nasser, and he was the student that drove this project. So we wanted to do a validation study asking the question whether it was feasible and valid to crowdsource systematic reviews. So we took 6 systematic reviews that were being completed by investigators here at GEO, and we pulled from those 2,500 citations with about 150 eligible or true positives. And then our goal was to have the citations assessed by a crowd we recruited online and by the expert teams. We wanted all the citations assessed at least 4 times, so 10,000 or greater assessments of title and abstract, and then we were anticipating 2,400 or more in full text. And we developed a website, which we called CrowdScreen. It's now shut down, but it allowed us, with a lot of work, to present the 6 systematic reviews to an online crowd. They could pick a review to participate in. There's a short training module associated with it, and then they could begin their work based upon the presented eligibility criteria. We promoted the study through emails and posters and word of mouth. Because the reviews were being completed by teams, we couldn't offer authorship to the individuals participating, but we could give them a gift card and a reference letter. So our first question we asked with this study would people do this? And we were quite surprised. We had about 200 people sign up, 100 of which were fairly active. Ultimately, they did 17,000 assessments of title and abstract. They retrieved close to 800 PDFs, and then they did full text reviews. There was over 7,000 assessments. So we thought that gave us a good sense that people would do this if a platform existed. And then the second question we asked was, what was the crowd performance compared to the expert? And the answer there depended a little bit on how you use the data. But example 1 being, if you combined all the crowd and only excluded if everyone said exclude, you had 100% sensitivity and work saved of about 60 to 65%. If you were a little bit less conservative and said, well, 75% of people would have to say exclude, you ended up with work sensitivity again around 100% and work saved in the 70 to 75% range. And then if you set up to 50% could exclude the article, you ended up with sensitivity still in the high 90s and work save of about 80%. So we're quite impressed with that. And we also asked the question, beyond the crowd, how did individual crowd members perform? And on average, the individual crowd member, which was largely academic in nature, had a sensitivity in the mid-90s and specificity in the mid-70s. So we were pretty happy with that. And we published that a couple of years later in the Journal of Medical Internet Research. So we were pretty happy that maybe we had a decent idea to help investigators do their systematic review. We had done some research suggesting it might be good, but we felt we were pretty far from the finish line. And the major barrier being that there really wasn't good technology that allowed for crowdsourcing of large systematic reviews. We were very fortunate that we were invited to participate in something called Hacking Health event that was hosted here in Ottawa at the Shopify centers. And there we were introduced to a group at the Algonquin College, the Office of Applied Research and Innovation. And the lead of one of the labs there had done some systematic reviews and remembered how difficult they were and offered to put a team of professors and students on this project. And that was back in 2017. And we've been working with them for about 3 years now to develop a citation screening software that also has the ability to facilitate crowdsourcing. So the basic citation screening software was completed in 2018, and then the additional crowdsourcing features were completed and tested as of the end of December 2019. And we've been using it locally at our research institutes for systematic reviews. So to date, we've had about 65 projects initiated, of which 10 have been large crowdsourcing projects, and 42 are completed. We've got about 400 users on the platform, and we've got about 250,000 assessments completed so far. And the team is working in 2020 to build in data abstraction and some project dashboards. So I'll give you some examples of some real world testing that we've done, some examples. So we also have a website associated with the platform on WordPress. It introduces the platform, what we're trying to do with it. It also allows us to promote events. So we've done about 10 events so far with a variety of scientific organizations, where they'll come with a moderate to large citation set size, and we'll set up a weekend or a week, and we'll promote it. People will come, read about the systematic review, read about the team, understand the goal, understand what they might get in return in terms of potential authorship, reference letter, knowledge, etc. Decide whether to participate, and then they'll log in and they'll do the test set, and if they pass the test set, join the team and help out. So our first review, I'll share the results of our first review, which was kind of fun. We were approached right when we just achieved functionality in 2019 by the Canadian Blood Services, an international collaboration for transfusion medicine, and they had a 3,000 citation review on albumin administration in sepsis. And so we set up a project where we wanted these citations assessed 4 times each, so that would be 12,000 assessments. So when we started promoting the event, and we had 74 people sign up and initiate the test set, of which 35 completed and passed the test set, and then the crowd proceeded to work, and it was fun to watch because the crowd proceeded to do 12,000 assessments in just under 36 hours. They had about a 5% conflict rate, and then they moved on, pulled in about 300 PDFs, and then proceeded to complete full text assessment in just under 12 hours. So just, all in one weekend, they're able to proceed through abstract and full text screening. And then 6 of the very active reviewers were invited to participate in data abstraction. So ultimately, when we looked at the data from this review, we had about 92% of the articles the crowd was able to manage, either include or exclude, without involvement of the PIs. So I'm not going to show every example we've done so far, but here was an even bigger project on urinary tract infections in young children that we did with the BC Children's Hospital. Was set up very similar, and again, they were able to complete all abstract screening and full text review in a matter of less than 2 weeks. This had 10,000 citations. And what was fun about this particular study was that their team came from 10 different countries across the world, as shown by the map put together by the PI. So that was fun and interesting. And then a COVID example. So as mentioned, I'm a doctor at a hospital. Back in February, March, there was a lot of concerns about PPE shortage, particularly as Canada at the time didn't manufacture any of its PPE, brought it in from other countries. We were really worried we might run low, and the question of whether or not it was safe to decontaminate masks came up. At the time, there was no guidance from Health Canada or the CDC on what to do around decontamination. So my research group embarked on a series of 4 systematic reviews looking at 3 methods of decontaminating N95s and then decontaminating surgical masks. And we were able to use the InsightScope platform and crowdsourcing to complete 4 systematic reviews in a month. The days to complete here is actually from protocol all the way to manuscript, not screening. We got slowed down a little bit, although 19 days isn't that slow by the fact that we were analyzing, writing up all 4 at once. These ended up having a much bigger impact than I thought. I initially planned on just providing this evidence to our hospital and region. And then we were approached by multiple government agencies in and outside of Canada about sharing these results. These manuscripts, because of their timeliness, were downloaded about 5,000 times from OSF, and all are now accepted for publications. And we have an ongoing living review that's being promoted by COVID Ed. If you want to see the living review, you can use this code here. So conclusions and next steps. So what we've seen over the past couple of years with our experience with systematic review crowdsourcing is it seems to be feasible and valid. We hope we've removed the technology barrier with the development of InsightScope. We've seen benefits to investigative teams, but in addition to benefits to investigative teams, we believe this opportunity benefits individuals, scientific communities, and society through an acronym we put together, scholarship, motivating, impact, learning, engagement, and just overall satisfaction. Next step for us is that we're hoping to continue to advance the technology. We're working on data extraction, abstraction. Right now, we're working with IBM on some computer-assisted abstract screening, embedding that in the program so we can do a mixture of crowd and machine for really large projects. And we're building in, we're trying to increase the social or team aspect of it with increased communications. We're also hoping to increase visibility. We're a new platform. As Neil mentioned, there's lots of platform out there, so we're trying to stick to large projects, crowdsourcing, communities, and not enter too much into the domain of other platforms. But we're looking at a website redesign. We're putting together some promotional videos, and we'll be presenting at some conferences. And we're inviting scientific communities to the platform to crowdsource projects. Thanks. There's my contact information if anyone wants to reach out. All the best.
Great, thanks so much, Dale. That was really interesting. We have one question that we have time for quickly from Matt Jones, who said, is there any gamification, introduction of game-like elements such as rewards, scoreboards, etc. in the crowdsourcing?
Yes, we have built in a few things. So each user has a profile page, and if you're participating in a review with someone else, they can access your profile page. And based upon the work you've done in terms of abstract screens, PDFs located, full text reviews, you do accumulate points, and we call it an InsightScope score. And then each project has a leaderboard, so you can look on a project and see how much work you've done relative to other people. And definitely that validation project I mentioned, that ended up being, it was, looking back, perhaps not that surprising, but at the time felt surprising how much that mattered. People really wanted to know how much they had done relative to someone else. Were they first, second, third for a project? So yeah, we are looking at introducing some gamification. We want it to not be overly competitive, but recognizing that competition can be helpful. Thank you for the question.
Thanks very much. That's great. We will now hand over to Sam Cheng, who has pre-recorded her presentation because it's an unearthly hour where she is. While Jess sets that recording up, Sam is a biodiversity scientist at the Center for Biodiversity and Conservation at the American Museum of Natural History. So over to Sam.
Good morning, everyone. This is Sam Cheng. I'm a biodiversity scientist at the Center for Biodiversity and Conservation at the American Museum of Natural History in New York City. Apologies, I could not be with you live today, but I'm looking forward to hearing your questions after this webinar. So today I'm talking about machine learning and open source technologies, specifically talking about some of the lessons we've learned from developing and running Colander, which is an open source, open access platform for evidence synthesis. So currently researchers depend on manual approaches for sifting through the piles of information and documents that we look through for conducting evidence synthesis. Unfortunately, many of these approaches that we currently use are quite inefficient. So in this case, the authors of systematic maps and reviews in environmental topics within the Journal of Environmental Evidence are reading on average 14,000 citations to search for what they need, ultimately only including about 2.4% of what they examined. So not only is this manual process inefficient, it's also quite labor intensive. So this raises significant resource challenges to ensure that reviews are properly staffed. Estimates of the cost of systematic reviews, for example, range anywhere from $30,000 to $300,000 US, and depending on the scope, can take several years, which can be a challenge when producing syntheses that intend to respond to decisions and challenges with relatively short timelines. So the reality of the synthesis process is that as the evidence base is constantly growing, reviews will need to be updated. So with such an onerous process, what we actually need is a dynamic way to actually be able to search, to filter, and to extract data to keep up with the pace of scientific research. The platform has several features. It's built on an open science principle, so the code is open source and is available for future community development. It was built to facilitate collaborative synthesis by teams. So it's online and it's free and allows for unlimited users per synthesis project. So while the origins of Colander are within the conservation sector, it works actually for any domain of knowledge. So we've built Colander to make the synthesis process faster, but also to ensure that the process remains transparent, and we continue to try and minimize bias in the process to ensure reliable syntheses. So Colander is actually a semi-automated system that retains user oversight for all final decisions on relevance or for sorting. Lastly, the output of Colander projects can be exported for use in other programs. We have an active community of users and many online resources on our community website. And for those that are interested, Colander's framework is built using open source languages and packages, which are listed here on the right-hand side, and more details can be found on our GitHub page. So I just want to take you through a brief walk of Colander and its functionality. Once you sign up for an account, you can set up a new review, which will have a homepage that looks like this. You can plan and organize your review up top, as well as put in your key terms, selection criteria, and extraction form that will inform the machine learning model. You will also see that your review is broken down into 3 stages: citation screening, from which the included articles will go to full text screening here, and then the final included articles will then proceed to data extraction, where you will label your documents. You can invite as many people to your review as you like and choose whether or not you want your documents reviewed by more than one person. So Colander uses machine learning in 2 systems. In system 1, the user imports citations into the platform. Colander then ranks these citations based on the presence of user-defined key terms. So these are their search terms. Users then go through these citations, coding them as included or excluded. And after the user has gone through at least 10 citations, The machine learning model reruns based on this input and re-ranks the list of citations based on relevance. So this is a dynamic, iterative process. So the more user reviews, the more accurate the relevance ranking will become. So specifically, what we do in this stage is we employ an algorithm called word2vec. And this identifies co-occurrences of words around your key terms that you see in bold here. So essentially, it reads the abstracts and determines relevance, and then users can choose to include or exclude the listed citations. So after users have screened their citations and come up with their final list of included document, so this is passed through full text screening, System 2 predictively labels articles based on user-defined categories. So for example, in this case, our user-defined categories are for labeling documents for a synthesis on conservation actions and impacts on human well-being. So users import PDFs, which are then converted into text. Then users must manually label 50 articles before the machine learning algorithm begins to predictively label the articles. In developing Colander, we found that because disciplines differ in how much semantic variation, so how much words differ, how many different meanings a single word or phrase might have, because of this variation that exists to describe different types of topics and categories, that a more universally useful methodology here was actually to display the sentences in the document that best predict the labels with confidence rankings instead of automatically sorting and labeling documents without any user oversight. So users would then choose the correct labels. And again, this is a dynamic process, so the model is constantly learning and refining the predictions. And then finally, the resulting data set and coding decisions can be exported for analysis in other programs. So you can see what this looks like in practice with sections of the text that Colander predicts are relevant for a specific label, and users can approve or reject those. So Colander currently has over 1,500 users. These are users who have about 1,200 active reviews in a variety of topics, for example, in conservation, environment, ecology, economics, and in medicine and education. So Colander's users are quite a diverse crowd. Some examples include the ISEAL Alliance, who's been using it to extract information on the impacts of certification, the American College of Physicians, who is interested in using the underlying algorithms to improve synthesis in medicine, and then within the Science for Nature and People Partnership itself, by providing a tool for ongoing working groups such as ecological drought, soil carbon, and ecological lovers for health to find and to synthesize evidence to inform research and practice. So one question we often get from Colander users is, when can we actually stop? Because this is an iterative, active learning, machine learning process, the tool does not automatically sort citations into included and excluded. So users are constantly going through a list of unscreened citations and sorting through them. So because Colander doesn't provide you with hard stopping points, as well, within the evidence synthesis community across multiple disciplines, not just environmental sciences, there's a little guidance on when we can actually stop screening. And when we look at the possibilities of what we might risk by stopping screening too early, or the resources that might be wasted by screening too many irrelevant citations, we can try and start to make decisions about what are appropriate stopping points by determining what the level of acceptable risk is for the type of decision that the synthesis intends to inform. So for example, thinking about, well, what is risk by missing evidence? How much evidence can be missed? And for us to still trust the process. And this will obviously depend on the type of decision, the urgency of that decision, the decision timeline, and various other factors. So while we can't provide you with any hard stopping points, what we can do is provide you with a tool to monitor your progress and track what is happening in your review in real time. So the Colander Companion is available as an open source R-Shiny application. And you can visit and input your review ID and your email address that's associated with that review. And you can do things like track your inclusion rate for your review. You can also do things like looking at your inclusion rate over a certain interval of total citations screened. For example, many reviews have been employing different types of metrics, for example, such as not being able to find, not finding, any relevant citations. After having screened a certain number of citations, they would deem that time to stop. You can also look at progress for individual users to see how many citations each user has screened. Calendar also has a feature where you can add tags to citations into full text as sort of preliminary ways to mark documents. And you can look at summaries of the tags applied for both included and excluded in all the citations that have been screened. As well, you can export bespoke data sets. When you use the export function in Calendar, it exports one set of columns of your data set. If you're interested in other columns that are not traditionally exported, you can choose to export them here. And you can also export your citation files. So your included or your excluded title and abstract, and as well as full text. And you can export that as a RIS file. So the second part of this talk is looking at what this means for building open source apps and what we've learned from the Calendar process. So on the one hand, because Calendar is open source, it provides us a really great opportunity for crowdsourcing innovation. So for example, Calendar community users have built add-ons and tools that complement and enhance Calendar's functionality. The second is the opportunity for Calendar to contribute to progress in machine learning and evidence synthesis. So by sharing this innovation, we built Calendar at a time when other machine learning softwares and platforms also existed, but those processes often weren't particularly transparent. And so what we aim to do was develop an active learning model that allowed for more user oversight. But this really is just the first step. There are many different ways forward for machine learning and evidence synthesis to progress. So we want what we have done to contribute to this. And so hence this is our commitment to an open source platform. But at the same time, this comes with many challenges. Calendar was built by a team of volunteer data scientists. So this team of 4 built Calendar in their own time, in nights and weekends, over the course of 18 months. So currently, one of our challenges is figuring out how do we actually maintain Calendar? How do we provide our users with support? How do we troubleshoot when the server goes down? How do we maintain hosting costs? And we've been able to do this through a combination of philanthropy and different types of funding strategies through our various institutional partners. But this is something that every open source software should be thinking about, is once you've built your tool, how will you maintain it in the long run? As well, another challenge is keeping up with developments. Software is always changing. Languages are always changing. Bugs pop up. There are new versions, people requesting, new developments to improve the tool. And so how do we do that when development time is costly at times and our tool is free to use? And this is something that we're still grappling with. One thing that Calendar as an open source platform has allowed us to really think about and to interact within the open source community around is to think about what the evidence pipeline could look like. So by creating a platform where there are interoperable inputs and outputs, they can come from other programs, they can be fed into different other programs, and trying to facilitate cross connectivity between these different applications, what we're actually seeing is an open source, a commitment to open source principles can maximize the potential of this entire landscape of tools that's emerging, rather than trying to build one do-it-all platform. And through our growing community of users, this network, through the evidence synthesis technology group, we're seeing a lot of potential for this moving forward. So with that, I will not be taking any questions, obviously, but I welcome any questions to be sent to my email or to the Calendar team email address. And please do feel free to visit the app, the community website, as well as the Calendar companion website. Thank you.
Awesome. Thanks very much to Sam for pre-recording that presentation. We don't have any time for questions and Sam's offline, but if you do have any questions, please pop them in the questions box for the next minute or so. And after this presentation, I'll actually be handing over to Jess again, just to close. I won't be presenting anything myself. But just to say thank you so much to everybody who's taken part for attending and everybody who's taken part in presenting. If you're interested in seeing this again or seeing the presentation files, please check the environmental evidence website for more details. So over to you, Jess.
All right, thanks, Neil, and thanks everyone for participating and sticking with us. That was a great session, lots of information. The recording of this will be posted on our YouTube channel, hopefully by the end of today. And there'll be an email going out to all of the attendees about that. I encourage you to join us for our next webinar in October. So the registration for that is open now, as well as for our November session. So I encourage you to go to our website, see what's coming up in the next few months, and sign up early. Thanks all, and have a great day.